Okay, so we are going to talk about the process of cloning. So cloning began in 1997 when Dolly the sheep was created. So first, let's define clones. So clones are organisms that have identical genetic matter where the sequences of bases in their DNA is the exact same. So for example, let's say we were to take human 1 and clone human 2. If we were talking about a specific gene in each organism, let's say human 1, the sequence for that gene is A, T, G, C, A. And its base pairing would be T, A, C, G, T. Now, if we were to look at that exact gene in its human 2 clone, it would be the exact same. So that gene base pairing would be A, T, G, C, A, and T, A, C, G, T. So what do you notice about these two? They are identical. So that's the idea of cloning. Now, cloning an entire animal from embryonic cells is the most common method of cloning. So if you have an embryonic cell is a cell that is undifferentiated. Now, as the cell matures, differentiation will occur where this cell will then make multiple cells differentiated into different cell types. So this cell type could be for nerves, this one could be for the stomach, and this one could be for eyesight. Now, the whole idea with cloning from embryonic cell is that these cells here have not become differentiated. So the whole idea is that you can then take these cells and create an entire organism. Now, the differentiated cells, since they do contain DNA for the organism, but through different things, you can only access the DNA needed for the cells for a specific function. Now, before the creation of Dolly the sheep, it was believed that once cells become differentiated, they could no longer generate new organisms. However, it was discovered that if these differentiated cells were then starved, which is called your G0 state, you can then take these differentiated cells that are starved and you can create a new organism. So in regards to Dolly the sheep, an egg cell was taken from a sheep and the nucleus was removed from the egg. Electric shock was then used to fuse the starved differentiated cell with a nucleus-free egg cell. So what you had a nucleus donor. You had an egg donor. Now, Dolly the sheep will be identical clones to this nucleus donor. So when you take your egg donor and you take your nucleus donor, basically you're going to have your egg cell. So we'll say that is an egg cell. And you will take your nucleus, and the nucleus will then be inputted into your egg cell. Now, obviously, in order for that to be fertilized, you're going to need a sperm donor, where then sperm will come in and fertilization will occur. Now, in order to get that nucleus from your nucleus donor into the egg cell from your egg donor, what needed to happen was electric shock needed to occur. So electric shock will basically allow a pore to form on your egg cell, and it will allow the nucleus to be inputted into the egg cell. So now, once this egg is then fertilized, what will then happen is it will be inserted into several sheep. So fertilized cell will go into multiple sheep. Now, in regards to Dolly, this egg cell was put into 277 sheep in total, where only one of them was able to form an identical sheep to the nucleus donor. Now, the whole idea of Dolly gave us an aspect to look at and say, we can then do this for animals. Of course, there is an ethical view to that, but Dolly the sheep gave us an opening to see what the future can hold for cloning and for humans itself.